Hello everyone, in this video we're going to talk about mutations. We talk about mutations not only because there are a lot of diseases that are caused by mutations. Everybody have mutations happening in their body all the time. It's just a part of the natural things that happen in the human body or any organisms. As a reminder, DNA can go through DNA replication, and then we have something called transcription that allows an mRNA transcript to be made, and then eventually translation allows a protein to be made. So there are a lot of parts of our DNA that actually codes for a protein or a trait, and we call that a gene. But there are also other parts of the DNA that doesn't code for RNA or protein, and you can learn more about it in AP Bio in a few years. So when we're talking about DNA to RNA to protein, ultimately DNA is the instruction that codes for the protein, right? So if there were to be a change in the DNA bases, that will lead to an mRNA change in the base, and there may or may not be a change in the protein. And if the protein is not functioning correctly, that could lead to some pretty serious complications in a human body, or really any organism at all, right? Because everybody has DNA, RNA, and protein. So a mutation, these are some of the questions that we went over in class. So we'll go right to the next slide. What a mutation is. So a mutation is a change in the sequence of DNA, right? Ultimately, as I said, DNA is what gives the instruction for everything. So we define mutation as a change in the sequence of DNA. Mutations can be beneficial, sorry about my dog, beneficial, harmful, or have no effect. So we'll talk about what are some of the mutations that will that are more beneficial, harmful, or have no effect. And then you want to know that actually most of the mutations, okay, back to work. So you want to know that although most mutations, although there are a lot of mutations, okay, most of the mutations actually have no effect, or by that I mean there's no negative or positive effect. So what we mean by that is because we said there are some DNA that code for proteins and some that don't, sometimes if you were to have a mutation in the region of DNA that doesn't actually code for proteins, then there might not actually be a consequence of the mutation. But there's something else that makes mutation not so harmful, which is called redundancy. So if you can remember, when we look at a codon chart, such as the one right here, you can see that there could be multiple mRNA that are coding for the same amino acid, right? So for example, if we look over here, if the original mRNA is supposed to be CGU and it codes for arginine, after a mutation, the mRNA could change to CG. Wait, did I say CGA? Anyway, CGU to CGA, right? So there could be a mutation that changes the base. You will still call that a mutation, but that doesn't actually change the amino acid that this mRNA is coding for, right? And a protein ultimately is, I'm saying ultimately a lot, a sequence of amino acids. And this sequence of amino acid is what determines how this protein is folded, and the shape of the protein is what determines the function of the protein. So if your amino acid is not changed, then this protein overall shape is also not going to change. And sometimes even if the amino acid is changed, if this amino acid is not crucial for the folding of the shape of the protein, then this mutation still is not going to lead to any kind of complications. Okay, so moving on. This picture is to show you the difference between a coding region versus a non-coding region of a DNA. So if you look over here, we have the DNA part, we have the exons, which are the parts of the DNA that is saved and is transcribed and translated, but the intron regions are the DNAs that are actually cut out before the mRNA is translated into a protein. So that part, although is a part of the DNA, it doesn't actually contain the information that makes the protein itself, right? So if there were to be a mutation right here, or right here, or right here, or right here, then that's not going to change how the protein is made. Then if this protein is what we're looking at, come here, Toby, then that's not going to change anything. Sorry, my dog is a little reactive to the outside world. So now we're going to talk about the causes of mutation. So a mutation is caused by a mistake made during DNA replication. Well, that's one way of causing it, right? So we said it's a change in the DNA basis. So if there is a mistake made during DNA replication, let's say you're supposed to have A and T pairing with each other, but then that didn't happen. An A and a G are paired together, right? Then that is going to cause the DNA to change, and that could lead to a change in the protein or not. So mistakes made during DNA replication is definitely one of the causes of mutations. However, there are also a lot of other environmental factors that can induce mutations, meaning that this could increase the rate of mutations beyond a normal, percentage. So for example, radiations, certain chemicals, for example, chemicals in cigarettes, or infectious agents. So there are certain viruses that would actually insert a piece of this DNA into your genome, and that is certainly not great, right? So one of the examples that I gave in class was called the herpes virus, right? Everybody hear about the herpes virus and they're so scared. But actually most of the human beings in the world carries the herpes virus because it's so easily spread and it stays in your genes forever. So how do you know if you have herpes or not? If we don't call it herpes, right? We call it cold sores. If you ever had those blisters on your mouth, we call it a cold sore, but really what causes it is that there are herpes virus that kind of stay hidden and stay dormant in your genes, and there are certain environmental factors or, change in hormones that can trigger these viruses to actually be made. And when these viruses are made, your body attacks the viruses, and the pussiness of when you're having a cold sore is actually your body fighting off the virus. So just putting it out there, everybody has herpes. All right, so there are different types of, don't focus on that, but there are different types of mutations, right? Different types of mutations, the big categories are called germline mutations versus the somatic mutations. So the germline mutations are talking about the germline, right? So mutations that happen in the gametes. And those are the types of mutations that can actually be passed on from parents to the offspring, right? Because you're only inheriting genes from the gametes of the parents, right? So that's a germline mutation. As you can see, if there's a mutation in the parent's gametes, then the entire embryo and the entire organism, every single cell in this organism is going to contain this particular mutation because your body is made from this original one cell, right? The division of this one cell. So germline mutation happens in the gametes. It can be transmitted to the offspring, and generally it's very significant of an effect, right? If you think about, there are certain mutations that causes a person that has not only change in the DNA, some people actually have an extra chromosome or one less or even multiple extra chromosomes. Then every single cell in this person's body will have that extra chromosome, and that creates problem in a lot of different ways, right? Moving on, the other one is called somatic mutations. So somatic mutation is easy for us to understand. It's mutations that happen in your somatic cells, so not in the gametes. This is something that's developed more, after the person is already born, right? So we have somatic cells, that's your body cells that are not the gametes, right? So then only a certain part of this individual is affected. So for example, that's my light turning on. So for example, in this picture, you can see that the shoulder region of this embryo has certain mutations, and obviously these cells are still going to divide, and when this person grow up, there's going to be a larger part of the human being that has this particular mutation, but it's still kind of regional. It's only going to affect the somatic cells of this person and not the gametes. And this particular mutation is not going to get passed on to the offspring unless it's something that's caused by the genetics itself. So that's more complicated. But remember, if you were to have a somatic mutation, generally you can't pass it on to the offspring. And generally this is less huge of a deal, but because in terms of passing it on to an offspring, it's not as big of a deal. But it could still have a lot of consequences for the individual himself or herself. Moving on, here are some review questions. You can look at that when the time comes. We're going to move on to specifically different categories of mutations. The first one is what we call a point mutation. Hold on, my dog is making holes in the ground. Here's my dog making holes in the ground. Anyway, this video gets stuck a lot for some reason. Okay, so point mutations, we can also call those substitutions. So what are we substituting? We're substituting one base in the DNA. The reason why it's called a point mutation in my head is because it changes one point on the DNA, just one base only. And this one change could have a pretty significant effect as well, but it's changing one base. So that's kind of the big category of this. It's called a point mutation. So there are a lot of words here. Let me break it down for you over here. Point mutation, change in a single nucleotide in the DNA, right? As a reminder, a nucleotide is a monomer for a DNA or RNA. If you're changing a nucleotide, you're changing one base. So you could change the letter A to a letter G, for example. So that's a point mutation. This only affects one codon because, as a reminder, one codon is 3 bases in the mRNA, right? So if you change a base in the DNA, that's going to cause a change in one base of the mRNA. And if you change one base only in the mRNA, that's only going to affect one codon in terms of the amino acid that this mRNA is coded for. So, however, right, sometimes mRNA codons can code for the same amino acid, so sometimes there's no sequence at all. So no consequences at all. So we'll talk about that. There's 3 different types of point mutation. That's kind of your subcategory. We have the silent mutation, missence mutation, and the nonsense mutation. And we'll talk about each one of them. The first one is called, well, actually, yeah, if you look at this codon chart, you can see that there's some mRNA codons that code for the same amino acid. So moving on, we have this example that we did in class. We'll just quickly look over that. So basically, you have an original message. That's your original mRNA transcript that's supposed to code for the protein. And then we have a certain mutation. And then you want to think about if there's a change in the letters. So this is kind of instead of amino acids, we're looking at letters, just as an example, where you want to see how this would actually affect a protein if this mRNA were to code for a protein instead of an English word. You'll get the idea, I think. So the first one is called a silent mutation, right? If you look over here, although there's a change in one base, A to G, right? This is a change in one base, so we can call it a point mutation or a substitution. This doesn't actually change the letters that it codes for or the actual amino acid that it codes for. So we call this a silent mutation because there is no real consequence to this mutation. There is no change in the amino acid, so there won't be any change in the protein that this mRNA is coded for. So that's called a silent mutation. The next one is what we call a missence mutation. So the same idea, it's the same cause. The cause is we're changing one base in the DNA, which causes the change in one base of the mRNA, right? This is still a substitution or a point mutation. However, the result of this mutation is that there is actually a change in the amino acid. Toby, come. Sorry. Making holes in the ground again. Okay, so when your point mutation actually causes a change in the amino acid, so if you think about it, if there is actually a change in the amino acid, there may or may not be a change in the protein shape, right? If this amino acid is very crucial for how the protein is folded, then that's going to be a pretty big deal. But if this amino acid is not as important for how the protein is folded, then even though the DNA is changed, RNA is changed, and the amino acid is changed, this protein could still be mostly fine. The last one, the last type of the point mutation is called a nonsense mutation. So this is a much bigger deal. This is the worst out of all 3. Okay, so if you look at a nonsense mutation, there is still, the cause is still the same. We're changing one base in the DNA, which changes one base in the RNA. So this is your original mRNA, and then we have a mutated mRNA, right? But you see that this word is shortened because instead of a change in the amino acid itself, this amino acid is actually changed into a stop codon. So the entire translation process is stopped. This is a huge deal because if you were to have a protein that's supposed to be, I can't stop holding onto my dog because then it'll run away, pretty long, right? So many dog problems. If you have a protein that's supposed to be really long and then this protein is cut short during the translation process because there's a premature stop codon, that is a huge deal. This protein is most likely not going to work at all because your protein is cut short, right? It's as if you had a key to a house and now your key is only half a key. That is not going to work out at all. So this picture shows you one more time how point mutation works. You just change one base. That's called a point mutation. But there are 3 different types of point mutation. We have silent, missence, and nonsense. We're going to look at it one more time. Silent mutation, you're still changing one base, right? But the result is it results into the same amino acid. So there's no effect at all. A missence mutation changes the amino acid, And it changes one amino acid only because we're only changing one base, which is one part of one codon. So the effect could vary. And then we have nonsense mutation where you have a premature stop codon, which means translation is cut short. Your polypeptide is cut short. This protein is not going to work at all. As you can see, a missense mutation, again, the cat ate the rat, the cat ate the hat. It's still pretty similar. It's not exactly right, but it's close enough, right? It's 3 letters at a time because that's how an mRNA is read, right? 3 letters at a time. A nonsense mutation, however, is going to change the cat ate the rat to the cat ate. And you'll never find out what it ate. So that's not great. An example of a substitution mutation is called sickle cell anemia. So this is only the change in one base, in one codon. And the effect is pretty big, right? The sickle cell anemia, I don't know how much you know about it. This is a missense mutation. It changes the original amino acid to a different one. But the effect is that the red blood cell of a person carrying sickle cell anemia is not in the right shape, and it doesn't carry as much oxygen as a normal red blood cell would. So it's a pretty big deal. But the cause of it is, it doesn't seem like that big of a deal if you were to just change one base out of millions of bases. What's wrong with that? A lot of things can go wrong. So then, in comparison to a point mutation, there's also something called a chromosomal alteration. That means you're changing, instead of only one base or one base pair, you're changing a larger part of the chromosome. So there's several types of this. We have a frameshift mutation, and there are 2 subcategories. One is called an insertion, one is called a deletion. And then the other bigger category is called a duplication. So this is one bigger category. This is parallel to that. These are subcategories of the frameshift mutation. And then we have the inversion and translocation. So we'll talk about each one of these as well. The frameshift mutation is the most important for you to understand. Okay? So insertion, here's how this works. Think about this. You're standing in line to get your Chick-fil-A sandwich. God forbid, somebody cuts the line in the middle, right? You're already at the back of the line. You're pissed off that you're not getting your sandwich. And then somebody cuts the line. And what's going to happen? Every single person after that person is going to have to move back by one step, right? You got to make room for that one person. So that's how an insertion works. Insertion basically is a DNA base cutting the line. It could be 1 base, it could be 2 bases, it could be 3 bases, it could be more. Either way, it's not great. Okay? So here is why it's not great. If you look at this example right here, our original message is supposed to be what with a question mark. And then the mutated message after this insertion is going to change every single letter after that, right? This, if you look at it, we added this one U base right here. Now, this A is going to move back by 1. U is going to move back by 1. Move, move, move. And why is this such a big deal? It's because when you read an mRNA, or when a ribosome reads an mRNA, it reads it 3 letters at a time. So if you were to insert 1 letter anywhere in the middle, then all the bases have to move back. And that means your reading frame is shifted back, right? So instead of AAA over here, you have AAU. Doesn't look like too big of a deal. But then the next one is UAG. Now it's changed to AUA, completely different mRNA, which means it's going to change the amino acid completely, right? And this next one is supposed to be UAG, but now it's changed to GUA. It's going to mess everything up, right? If there's an insertion, every single amino acid after the insertion is going to be messed up, right? So as you can see, this word is completely messed up. That's an insertion. But if you think about it, inserting 1 versus 2 versus 3 bases, or 3 or 6 bases, 3 bases insertion or 6 bases insertion is not as big of a deal, because instead of shifting the entire reading frame, you're simply adding an extra 1 or 2 amino acids. So it's not as bad. Hope you understand that. Please come see me if you don't. Right? So if you look over here, showing you the same thing. If we were to add this 1 extra C, every single base after that gets pushed back, and your reading frame, your 3 bases at a time, right? This reading frame is now wrong because everything is shifted. This one is also wrong. Every single thing is wrong. Now, same idea. Insertion is bad. Deletion is also bad. Although if you're standing in a Chick-fil-A sandwich, you wouldn't be too pissed off if someone decides to leave. That doesn't usually happen. But if it did happen, right, somebody decides that they would rather eat some Popeyes instead. But if you were to have a deletion, that means everybody have to move up by one step, right? But a deletion also changes the reading frame because you're reading 3 letters at a time. And if you're missing a letter, every single letter has to shift forward, or every single base has to shift forward, right? So as an example, you have this original message which codes for protein, but now you get rid of C and A. So instead of getting rid of 1 base, you get rid of 2 bases, which is just as bad. Now you have who knows what this is, right? So this one is easier to see. You can see that after getting rid of C and A, 2 bases, every single one of the bases after that is going to shift forward by 2. And now this one is messed up, this one is messed up, this one's messed up. Every single we call codon. Oh my gosh. Every single codon is messed up now. Okay, so that's not great. We call, hold on. So as you can see, if we get rid of this 1 base, every single base shifts forward, and the reading frame, right, is shifted. That's why you call it a frameshift mutation. So insertion and deletion are both types of frameshift mutation. Okay, now, if you look at the notes, insertion and deletion involve inserting or deleting a certain basis, and you can call that a frameshift mutation and because all the codons downstream, right, if you're looking downstream afterward, every single one is going to be affected. That means your protein is definitely not going to function because most of it is wrong. Most of the polypeptide, part of the polypeptide chain is simply wrong, and that's not great. This is called a frameshift mutation. If you look at the bottom example, you can see how a deletion works, how an insertion works, or if you were to read 3 letters at a time in a sentence, the fat cat sat, and you get rid of T, and you still read 3 letters at a time, none of these words make sense, and that's how it works in a protein. None of the amino acid is going to be right after the insertion or deletion. There's a lot. We're almost there. Okay, here's some examples of frameshift mutations. Now we're almost done. 3 more. Other types of chromosomal alterations: duplication, inversion, and translocation. So duplication is simply there are different parts of the chromosome, right? Long DNA, different parts of the chromosome. If there's one part of the chromosome that duplicates, that's called a duplication. That should make perfect sense. My dog is kicking me with his dirty paws. Okay. Okay, so for example, if you had this particular mRNA sequence and the codes for, if it were to be the pseudocode, it would have been Oscars, and you duplicate a certain part of it, now you have Oscars. The effect of this, it could be pretty bad, but it might not be that bad. We'll have to see. So that's called a duplication. And then the inversion is there's a part of the chromosome and it flips 180 degrees. The first base becomes the last base, second to the first becomes the second to the last. Every single base goes the opposite. That's called an inversion. Okay. For example, this is what you see, but it doesn't look as good as the duplication because with this part, since you're flipping, right, you're not just switching the 2 codons, you're flipping every single base, then the effect is a little bit more, right? This doesn't look like bubbles anymore. That's called inversion. And lastly, we're on to the inversion. Okay, you can see how it switches 180 degrees. Last one is called translocation. So translocation has to involve 2 chromosomes. It's almost like your typical, whatchamacallit, hold on, I'm blanking. When you have 2 homologous chromosomes and you have the crossing over, remember? If you have 2 homologous chromosomes, one part crosses and then it switches. It's kind of like that, except for translocation, it doesn't have to be homologous chromosomes and it doesn't have to be the end of the chromosome. So you have 2 random chromosomes anywhere inside the cell, 2 parts of this chromosome can switch. That's called translocation. Okay, so it involves 2 words. Okay, and the effect is not great. Okay, that's all we have. But now I want to go over one question on the homework so you know what we're talking about. I think a lot of you are confused, but I did want to put that challenge out there so that we can talk about it in class tomorrow. So now that you just watched this video, you should know what a missense mutation is, right? So we're going to use that as an example. A missense mutation is when you change one DNA nucleotide, which changes one mRNA nucleotide, and it changes one amino acid. So that's a missense mutation. It changes one amino acid, right? So how can you come up with an example? I think that's going to be the more difficult part for you. So let's take a look. We have our, okay, so let's take a look at how this will work for a missense mutation. So we have our original DNA, which codes for a certain RNA, and then the amino acid sequence. But if you think about a missense mutation, right, the point is an amino acid does change as a result of something changing in the DNA and RNA, and you can only change one base. It's kind of like figuring out a puzzle, kind of backward, right? But if you are able to give an example yourself, that means you're truly understanding how this mutation works. So let's take a look. Methionine is hard to change because there's only one thing that codes for methionine. So we'll move from that. Let's look at glycine, right? If you look at glycine, in your original mRNA, you have GGU, which is your codon that codes for glycine. And you can see on the codon chart that GGU mRNA does code for glycine. Great. This is exactly what we wanted originally, right? However, now you want to look for a change in the base. You want to change GGU into something else that will give you a not glycine. How would that work, right? If you think about it, you have GGU, and you can look on the amino acid chart. What if we change GGU to UGU? Why not, right? GGU to UGU, you're changing one base. That's fine. Does it give you a change in the amino acid? Absolutely. It changes from a glycine to a cysteine, right? So what you can write here is your mutated amino acid sequence will now be met, cis, and lu, right? Because you're only allowed to change one base, right? Going backward, you would have AUG instead of GGU. Now you're going to have UGU because that's how the mRNA could change, right? And then you have CUG. Everything is fine except this one base right here. And then going backward one more time, how would that affect our DNA, or what kind of DNA would cause this kind of change in the mRNA, which causes the change in the protein? So your original DNA should have been, or your mutated DNA should have been TAC, and then instead of CCA, you would have ACA, right? ACA versus CCA, that is change in one base of the DNA, exactly what a point mutation is. And then the last one should be GAC because we're only changing one base. So we want to keep the last one the same. Okay, I hope that makes sense. Let's try one more. Maybe let me give you one more example. So methionine, glycine, leucine, right? What if you want to change leucine instead of the other two amino acids? So the original one you have is CUG. You find CUG on the codon chart, and that's leucine. Great. Your goal is to change one base in the mRNA that actually allows the change of this particular amino acid, right? Because that's what a missense mutation is that you're changing one base of DNA, which changes the base of the RNA, which changes the base, changes one amino acid. And that's what you're trying to do, right? So originally it's leucine, and then instead of CUG, let's see what we can change. What if we change it to UUG? That's not great. It's still leucine, right? So you don't want that. What if we change it to AUC? There we go. So AUG. How about that? AUG. Right? So originally you had CUG, you changed one base in the RNA, now you have AUG, so now that changes it to methionine. So now your original amino acid, your mutated amino acid sequence, instead of methionine, glycine, and leucine, you would have methionine, glycine, and methionine. And your original mRNA is going to change to AUG, GGU, and instead of CUG, you're going to write AUG, because that's what causes this leucine to change into methionine. And then your original DNA is going to mutate into TAC, CCA, and TAC, right? So ultimately, the change from this G base into TAC is going to cause the change in the mRNA. Instead of GAC, now you'll have AUG, and instead of leucine, you'll have methionine, right? So it goes both ways, but in this particular practice, you're trying to go backward, and you're looking for a specific result. You're trying to figure out what would cause this result, okay? I hope this video makes sense. I hope you're watching this, and I apologize for my dog. My dog's weird behavior. Might be too lazy to actually cut out those parts, so I'm sorry.